Hello, and welcome to another episode of Fishing with Ashlee. My name's Ashlee, and today's webinar focuses on the NTRK genes and the clinical applications associated with them. It's important to note that while we are probe developing and fish testing experts here at Empire Genomics, we are not research scientists studying the NTRK genes. This webinar is presented to you in an informational manner to help you understand the clinical history of the genes, the fusion partners of the NTRK genes, and the clinical applications. So with that being said, without further ado, let's jump into this webinar. So starting off with gene fusions, the push towards targeted cancer therapy has established gene fusions as a targetable class of genetic mutations. Targeted treatment has generated survival rates far higher than those achieved with standard chemotherapy. And the NTRK gene family is associated with fusions relating to a variety of different conditions. And the NTRK gene family consists of the NTRK1, the NTRK2, and the NTRK3. And the encoded receptors are TRK-A, TRK-B, and TRK-C. The neurotrophin receptor tyrosine kinase, or the NTRK family, is made up of 3 genes crucial to the nervous system development and physiology. And each receptor is activated by a different binding factor, which triggers phosphorylated of the protein's intracellular kinase domain, setting off various signaling pathways that maintain normal nervous system function. So taking a deeper look into the NTRK1, this is located on chromosome 1, Q21 through Q22, and the encoded protein is the TRK-A. This gene is expressed in specialized neurons of the basal forebrain that monitor memory processes, pain, and temperature sensing. TRK-A contains an intracellular domain containing a juxtamembrane region, a TK domain, and a short C terminal tail. TRK-A serves as a receptor for nerve growth factor beta, or NGFB. Upon binding with NGFB, TRK-A phosphorylates itself and other MAPK pathway proteins. Looking at the NTRK2 gene a little bit closer, this gene is located on 9q22.1, and the encoded protein is the TRK-B. So the TRK-B receptor binds to brain-derived neurotrophic factor, or BDNF, and neutrophin 4. So BDNF is primarily associated with production upon exercise. It kind of aids in mood stabilization, neuroplasticity, making new connections in the brain. So the TRK-B expression is highest in the prefrontal cortex, the amygdala, and the occipital lobe. The gene promotes neural cell plasticity, again, neuroplasticity, and survival, as well as synaptic signaling and possibly even mood stabilization, again, released during exercise, stabilizing the mood. TRK-B also assists in learning and memory by aiding both short-term synaptic function and long-term potentiation. So looking at TRK3, this gene is located on chromosome 15, Q25, and the encoded protein is TRK-C, or TRK-C. TRK-C is a glycoprotein primarily expressed in the hippocampus, cerebral cortex, and the granular layer of the cerebellum. The NTRK3 mediates neuronal differentiation and survival via activation of the MAPK and the PI3K-AKT pathways. The gene may also aid in proprioception, or the body's awareness of where it is in space, and heart development. So just a brief history of the NTRK here and the research behind it, as well as the treatments associated with it. So back in 1982, the NTRK fusion was identified in a colon tumor biopsy. Moving through to 1998, the first ETV6 NTRK3 fusion was identified in infantile fibrosarcoma. Looking at 2010, we have the NTRK2 fusion identified in pilocytic astrocytoma, which is the most common childhood brain tumor. And then looking at 2015 through 2019, you can scan through if you'd like. They're associated with treatments that have been developed due to NTRK fusion cases in small cell lung cancer, colorectal cancer, neuroblastoma, solid tumors, and more. So I'll give you just a moment to briefly look over this if you'd like to, and look over the associated drugs that are suppressors of these genes. So let's just take a moment to understand gene fusions. And to the left here, you can see 3 slides with beautiful images of gene fusion probes. So fusions indicate translocations of genes. 2 genes that are initially separate translocate and end up fusing together. If the results of the fish testing show 4 separate probe signals, for example, 2 orange signals and 2 green signals, a fusion has not occurred. If the results show 2 fused signals and 2 individual signals, for example, 1 orange signal, 1 green signal, and 2 fused orange and green signals, this indicates a translocation and a fusion of the targeted genes. Another positive fusion result may look like 2 individual signals and 1 fused signal, and that is 1 orange and 1 green signal, as well as 1 orange and green fused signal. So we're going to take a look a little bit deeper into the fusion partners of these genes and looking at NTRK1 fusion partners and the diseases associated. First, we have the IRF2BP2 gene fusion, and this is associated with some lung cancers, thyroid cancer, and prostate cancer. Looking at the fusion with the LMNA, there's obviously quite a list here, but a few just to touch on some are gallbladder carcinoma, breast cancer, colorectal cancer. Fusion of the NTRK1 with TPM3 has been found in breast cancer, cervical cancer, colorectal cancer, amongst others. You can see the list there. And the fusion of the NTRK1 with the TPR has been found in lung cancer, thyroid cancer, uterine sarcoma, and pediatric mesenchymal tumor. Looking at fusion partners of the NTRK2, and again, these are just partners that have been found through research and are associated with these diseases. So fusion of NTRK2 with the DAP21P is associated with colorectal cancer. Fusion of NTRK2 with NOS1AP is associated with anaplastic astrocytoma and glioma. Fusion of the NTRK2 with the SQSTM1 is associated with glioma and lung cancer. And finally, fusion of NTRK2 with the TRAF2 is associated with melanoma. Looking at some NTRK3 fusion partners, first one being EML4, that is associated with congenital mesoblastic nephroma, glioma, infantile fibrosarcoma, and thyroid cancer. Looking at NTRK3 with the ETB6, and this is a more common fusion that a lot of clinical labs are associated and aware of. There's quite a long list of cancers here, but just to name a few: acute myeloid leukemia, breast cancer, colorectal cancer, glioma, infantile fibrosarcoma, lung cancer, melanoma, and the list goes on. And then NTRK3 fusion with the RBPMS is associated with thyroid cancer and uterine sarcoma. So cancers with the highest prevalence of NTRK fusions, the first one being secretory breast cancer and salivary gland carcinomas. Secretory breast carcinoma have the ETB6 NTRK3 fusion with over 90% frequency, as well as the salivary gland carcinomas. They tend to display the ETB6 NTRK3 fusion in more than 90% of cases. And it also notes that ETB6 NTRK3 fusions have also been detected in secretory carcinomas of the skin and thyroid. Infantile fibrosarcoma is an extremely rare childhood tumor, and the ETB6 NTRK3 fusion has been shown to occur between 70 and 91% frequency in these tumors. The LMN1 NTRK1 fusion and the EML4 NTRK3 fusion have also been associated with infantile fibrosarcoma. Congenital mesoblastic nephroma, this is a rare tumor that occurs in the kidney and is most commonly found in newborns or young infants. And the ETB6 NTRK3 fusion are found in these types of tumors, and the fusions typically occur alongside a trisomy 11. Thyroid carcinoma. Thyroid cancers in patients younger than 20 years of age account for more than 2% of all thyroid malignancies diagnosed in the US. Of these diagnoses, papillary thyroid carcinoma makes up about 90% of those cases. And in one study of 27 pediatric PTC or papillary thyroid carcinoma patients, NTRK fusions were found in 26% of the cases. And then NTRK fusions are also found in adult thyroid cancer patients, but at a lower frequency of around 10%. Brain tumors. So in a study of 112 pediatric high-grade gliomas, NTRK fusions were detected in 8 of the patients and were more common in non-brainstem high-grade glioma patients of less than 3 years of age. So NTRK fusions have also been detected in breast cancer, non-small cell lung cancer, colorectal cancer, and melanoma. So NTRK fusion frequency does not exceed 5% for these types of cancers as of now. However, as widespread screening for NTRK becomes more common, the incidence of these fusions will likely be better reported. So in the coming years, we'll achieve a more accurate idea of the true prevalence of NTRK fusions across these major cancer types. So studies have already supported that NTRK fusions are prevalent in a variety of conditions and cancers, but the door is really open to continue researching these genes and better understand their prevalence in certain cancers. Ongoing NTRK research. So despite the success already achieved with current targeted treatment, much work remains to be done in researching NTRK genes. There are several clinical trials currently in place investigating the efficiency of various TRK inhibitors. So if you want to keep up on the latest NTRK research, you can visit this website listed here, and I'll give you a moment to write that down if you're interested. It's clinicaltrials.gov. So this final slide here is just depicting the ideograms that Empire Genomics has of the NTRK break-apart probes. So these probes are really great for determining if the NTRK gene has translocated and fused elsewhere. Empire Genomics also offers fusion probes for the NTRK genes, such as ETV6 NTRK3 fusion probe. If you want to learn more about Empire Genomics fusion probes or any of Empire Genomics probes relating to NTRK fish probes, you can visit empiregenomics.com/NTRK.